genitals, also referred to as the external nares, a rather large mouth with a protruding tongue. On the body of the fetal pig, observe that there are two pairs of appendages. On the forelimbs, we can see that the hooves form the digits. Also, we can see the presence of a wrist, elbow, and shoulder. On the hind limbs, observe that there is an ankle and a hip. If we take a look at the abdominal wall of the fetal pig, we see the presence of the umbilical cord. The umbilical cord is what connects the fetal pig to the uterine wall of the mother and is present throughout the fetal lifespan. Within this umbilical cord, there are two umbilical arteries, which are shown here in pink, and one umbilical vein. At the posterior end of the organism, immediately under the tail, we see an opening, which is the anus that removes solid waste. In sexing your fetal pig, check for the presence of a urogenital opening immediately below the anus. If there's one present, that tells you that you have a female. Also in this area is a urogenital tubercle. Looking at the male fetal pig, we see the presence of a swelling at the posterior end known as a scrotal sac. In the adult male pig, this will house the testes. Also note that in the male pig, immediately below the tail is an opening called the anus. The urogenital opening of the male is just behind the umbilical cord. And if we follow this caudal towards the anus, this is a penis. In both the male and female fetal pig, note the two rows of mammary papillae. In the next section, we will begin the dissection of the fetal pig.
Does everyone have the 3 to 4 structures?
3 to 4 structures? Yes.
How do you not get fetal?
Lag.
The boobs, the nips, ankles,
Section 2 mechanisms.
This is the section you do not have to write. No observation for making decisions. This is just for your own education.
We're now ready to begin our dissection of the fetal pig. Place your specimen dorsal side down in the dissecting tray. Make sure that you tie down both pairs of appendages. Once they're securely tied, we're ready to make our incisions. Start up at the base of the neck at the hairy papilla. Note on the side here there is a rather large incision that was previously made to inject dye into the veins. Starting at the hairy papilla, make your cut so you're just cutting through the skin and the muscle tissue. Take care not to go too deep so you won't damage any of the internal organs. Once you get down to the umbilical cord, cut around the cord on both sides. In the female fetal pig, you're going to go right down the midline, caudally. However, in the male pig, if we do this, we damage the penis. So what we're going to do at this point in the male is cut to each side so as not to damage the penis. Once you've made your initial incisions, go back through those a little bit deeper so as to go through the body wall. In the pectoral region, you'll have to add a little additional pressure to cut through the sternum, otherwise known as the breastbone. Once you've cut through the body wall, make lateral cuts just anterior to the hind legs on each side. Also make lateral cuts just caudal to the forelimbs. Once you've made these lateral cuts, you're able to fold back these flaps of tissue. And as you do so, you're going to have to cut through this partition between the thoracic and abdominal cavity. This partition is made of muscle and is known as a diaphragm. Cut through the diaphragm then. Fold back both flaps of tissue. And note that you're going to have quite a bit of preservative in here and maybe some brown fluid. At this time, you want to take your specimen to the sink and flush out this fluid, and then you are ready to observe the internal anatomy.
Okay. So a diagram of that incision is in your lab manual, and it's that figure. Okay, so if you had fetal pigs, this is the incisions that you would make.
So that's procedure.
That would be just making incisions. Yeah.
Is that what's the method part?
No. You can just mention it, that they use a dissection method, but you don't have to go into details of what you do.
Can you make a picture into this?
Yo, y'all need to get some bigger. I got your puny ass phone. Touch me again, see if I don't fuck you up. I'm gonna tell you that right now.
We're now ready to begin our study of the internal anatomy. Note this blue structure emerging from the umbilical cord is the umbilical vein. We have tied the umbilical vein in two places with a piece of string. We will now sever the umbilical vein between the two pieces of string, and this way we have freed the umbilical cord. The body cavity of the fetal pig is divided into two cavities. That which is cranial to the diaphragm is known as the thoracic cavity. That which is caudal to the diaphragm is known as the abdominal cavity. In studying the thoracic viscera, we can see up here in the neck region two very large glandular structures known as the thymus gland. The thymus gland also extends partially over the heart. Also in the neck region, we see a reddish-brown bean-shaped structure, which is referred to as the thyroid gland. This structure aids in metabolism. Also in the thoracic cavity, we see the presence of the heart and a pair of lungs. In the abdominal cavity, this large reddish-brown structure is known as the liver. It functions in producing bile, which will be used in fat digestion. Directly underneath the liver, we see this greenish sac-like structure, which is known as the gallbladder. It stores the bile until it is secreted into the intestine. On this side, underneath the liver, we see this large sac-like structure, which is the stomach. The stomach stores food and also chemically and physically digests it. Here in this curve around the stomach, we see this long reddish-brown structure, which is known as the spleen. The spleen functions in destroying old red blood cells. If we lift up the stomach and the spleen, we can see underneath this structure a granular organ known as the pancreas. The pancreas functions in producing digestive enzymes, which will also be used in digestion. Over to this side, we see a mass of coiled tubules. These are known as the small intestines. This is where most of the chemical digestion takes place and the absorption of the digested food. On this side, we see a compact coiled structure, which is called the large intestine or colon. It functions in storing the undigested food, which are also known as solid waste, and absorbing excess water. If we lift the large intestine, we can see a pouch where the small intestine and the large intestine join. This pouch is referred to as the cecum. If we follow the large intestine down caudally, this tubular structure is called the rectum. It serves in removing solid waste from the body. This will continue caudally until it reaches the anus, the external opening. Here we can see the umbilical cord again. The umbilical cord has two umbilical arteries which will emerge. They go on either side of the sac-like structure, which is the urinary bladder. The shiny material which lines the body cavity is called the parietal peritoneum. The other type of peritoneum in the body covers the organs. This type is referred to as the visceral peritoneum. In our next section, we will study the mouth cavity.
Okay, so just a note about what you're gonna put down for your methods. Notice that she's using a poker to identify the structures. That you can put in your methods. It's not just she opened up the pig and pointed. She actually used the tool to identify the structures.
It's called the poker?
Yeah, because I forgot the name of it. We're gonna call it a poker.
So it'd be like use scalpel to dissect this pig and use poker to identify structure.
Okay, so everyone understand that for your methods?
Yes.
Methods, procedure.
Use the scalpel. She used the scalpel to cut it open with a poker to identify the structures.
Like cards.
I don't know if you just said.
6 and 4.
We're now ready to begin the study of the mouth cavity. To expose the mouth structures, make a slit in each corner of the mouth. Then, drop the lower jaw to expose the mouth structures.
This is so freaking disgusting.
Observe the tongue, which has numerous little bumps called papillae. These papillae contain the taste buds, which function in taste. On the roof of the mouth, observe the teeth that have already formed. These will help in physical digestion. Also forming the roof of the mouth are two structures. The anteriormost structure, which is made up of bone, is called a hard palate. Caudal to the hard palate is the soft palate, which is made up of a membrane. If we look down into the throat area, which is also called the pharynx, we can see two openings. One opening leads down to the esophagus, which will go to the stomach carrying food. The other opening, which is called the glottis, leads to the trachea carrying air. Covering the glottis is a flap of cartilage called the epiglottis. This epiglottis will prevent food and liquid from going down into the trachea. In the next section, we will study the urogenital system.
Wow, that was really short.
Everyone got their 3 to 4 structures?
Yes.
So, just a question. What's different about a baby pig after it's born than a human after it's born? Based on what you find in the mouth
We don't
cavity.
have,
The
we already have
teeth.
That's crazy.
We're now ready to begin our study of the urogenital system. Note that I've removed the small intestine and the large intestine with the exception of the rectum. I've left the rectum intact. Also, much of the tissue in this area has been removed to expose the reproductive and excretory structures. The urinary or excretory system is structurally related to the reproductive system. As a result, we'll study these two systems together. Up here, in the dorsal wall of the abdomen, we see a pair of kidneys. These kidneys are covered with a tissue called peritoneum, as a result, referred to as retroperitoneal. The peritoneum is still intact on this kidney. On this kidney over here, I've removed it to expose the kidney.
Duffy right here.
On the medial border of the kidney, there's a tube emerging from the kidney and extending caudally. This tube is a ureter. It will function in carrying the excretory waste filtered by the kidney down to the bladder where that urine will be stored. Note that the bladder is a large sack-like structure that is sandwiched between these two umbilical